Hello, everyone. My name is Mingshuo Liu, and today I'm excited to share my work, the FlexiDrug, a domain-informed framework for precise and stable conversational drug editing. So, the drug editing refers to the iterative modification of molecular or protein structures to achieve therapeutic properties. So, just like the two images here to share how we can add more structures, functional structures into the small moleculars or the proteins. So with the rise of a large language model, it's like Llama, that is we can locally deploy it. We now have new opportunity to automate and optimize the drug design while ensure the data security. So we have the core of our FlexiDrug is conversational drug editing. So the users can interact with the model to modify the molecular structures. For example, if we user we have input, we can input any, for example, small molecular. We design this molecular to be more soluble in water. How can we do that? We're going to transfer this message into our PDDS, that is the prompt designed for domain-specific knowledge. So they can translate our request into a structured prompt, so cooperating with molecular knowledge. Then it will process by our Llama 2, our local deployed large language model. But there's a question how we can ensure the output is valid. Uh, we bring two approaches. First, we can enhance the Llama model using over 1.4 million small molecular instructions and over 500K uh, protein-based instructions to teach it chemical and biological principles. And also we have another model that is the retrieval database and the domain feedback. So domain feedback is going to determine if the drug we generated is valid. And we have a retrieval database that is if the generated molecular is invalid. We can retrieve the similar valid moleculars and ensure they still meet the original goal. And then our iterative process going to continues until a correct molecular is found. Uh, if the generated molecular is still not valid, we just input back into the IEDF and do the retrieval process again. So this framework allows our FlexiDrug to generate the precise and stable molecular modifications. We can support both small moleculars and the protein structures. So here is we can share our performance for the small molecular first. We tested on the single property and the dual property editing on small moleculars. So we can clearly see, okay, our Llama 2 that in our FlexiDrug is clearly beat the Llama 2 in other that is no domain knowledge informed the framework. And even we have several tasks that can like even beat the ChatGPT Turbo. So that's really good performance. And also we for small molecular we now using selfish-based output and instead of the smiles. And that brings us a lot of more like valid generation because the selfies can naturally guarantee that generated the small molecular expression is valid. Also, we have performance evaluation on the protein. We have two tasks. So for 501, that is for more alpha structures, and for 502, that is the beta structures. So we can find our FlexiDrug that based on Llama 2 that did a very good job, especially on the 501. It beat the ChatGPT turbo-based modification over like 30%. Also, for the visualization of our output, we can clearly say, okay, the protein sequence after we just visualize it's a 3D structure, it just bring us more alpha and beta structure there. And that would be it. Thank you for listening.